We have a highly detailed map of Alzheimer's disease at its beginning, defined by immune cells losing their regulatory balance. We have an equally clear picture of its end stage, defined by a total collapse of the brain's synaptic architecture. But between those two states lies a mechanical blind spot. There is a creeping physiological shift that bridges these two isolated conditions. Historically, the clinical field has treated this long middle phase as a diffuse systemic decline. It is often viewed as an inevitable general deterioration rather than a specific event with a distinct location. The passage from immune disorder to synaptic collapse is actually driven by the failure of a single structural address. Identifying this address allows the structural threshold where immune failure becomes circuit failure to be mapped, timed, and intercepted. That structural address is the perineuronal net, or PNN, a condensed reticulated mesh of extracellular matrix that surrounds the bodies of specific neurons. Standard biological models currently treat this net as an incidental victim of the disease. It is seen as downstream scaffolding that simply degrades as the local environment becomes toxic. The perineuronal net functions as a protective pivot, shielding the cell against its own metabolic and oxidative throughput. Stripping away this specific extracellular armor is the proximate lesion that actively pushes the disease into its final phase. Alzheimer's crosses from a disorder of immune regulation to a disorder of cortical computation precisely when this net is stripped, a structural event that initiates the transition toward synaptic collapse. To understand how this structure dictates survival, we can look at a natural control experiment within the cortex itself. Inhibitory neurons do not die on the same timeline as the disease progresses. The somatostatin interneuron is an early casualty. It faces its chemical environment unarmored, operating without a perineuronal net. The parvalbumin interneuron is heavily shielded by a dense aggrecan lattice. It fires at high frequencies, exposing itself to oxidative stress. The net sustains this by buffering toxic cation fluxes. As disease progresses, the unprotected somatostatin cell vanishes early. The shielded parvalbumin cell survives late. The timing of inhibitory failure follows the status of this net. The unprotected cell falls when the environment turns, while the protected cell survives only as long as its shield remains intact. The assault on that armor begins when lipid-burdened microglia shift their behavior. They stop secreting regulatory cytokines and release matrix-degrading enzymes. Specifically, ADAMS-TAMPS proteases, enzymes that directly target and dismantle the parvalbumin cell's protective net. This introduces a major paradox in the clinical literature. Some post-mortem studies show these nets destroyed by the disease, while others insist the nets are perfectly preserved, even in late-stage tissue. We can resolve this contradiction by looking at the dual structure of the net. It relies on a heavy structural proteoglycan core made of aggrecan, which is surrounded by a thin, decorative sulfated glycan coat. Standard laboratory lectin assays only stain the sugar coat. They are functionally blind to the structural core underneath. Microglial proteases cleave the critical structural core, destroying the net's protective capacity. But this chemical cleavage often leaves the superficial sugar coat physically suspended in place. Researchers reporting preserved nets in late-stage Alzheimer's are photographing structural phantoms. The armor's protective integrity fails long before standard assays can detect its absence. Once the microglial attack degrades the structural core, the parvalbumin cell is suddenly exposed to its own extreme metabolic throughput without a buffer. To avoid poisoning itself with unbuffered oxidative stress, the cell executes a desperate survival strategy. It ceases its fast spiking activity and halts its expression of parvalbumin proteins. The neuron powers down to a quiescent state. It averts immediate cell death by severely restricting its own function. This creates what we call the latent interval. There is a prolonged gap in time between the loss of the extracellular net and the ultimate death of the neuron. Standard clinical tools scan for electrical activity and parvalbumin expression. When they find neither, they misinterpret this silent adaptation as complete cell loss. The silenced cell is a casualty in waiting. Because it is still physically alive, this interval represents the disease's most consequential window for medical rescue. The consequences of this silenced interneuron extend outward, directly impacting the broader network of excitatory pyramidal cells. The first channel of damage is disinhibition. Without the parvalbumin cell pacing the local circuit, surrounding pyramidal cells instantly become hyperexcitable and hypersynchronous. This forces pyramidal cells to manufacture and eject massive quantities of toxic amyloid and tau particles into the local environment. The second channel is direct loss of exclusion. Subsets of excitatory neurons also possess perineuronal nets. When those nets are stripped away, the cell loses its physical barrier against tau internalization. The third channel is synaptic destabilization. Stripping the matrix scaffolding reopens a juvenile-like plasticity in the brain, physically destabilizing mature, consolidated memory circuits. The removal of the net actively conscripts the excitatory cortex, forcing it to become an engine of widespread, accelerated pathology. We can now trace a direct mechanical line. Matrix loss causes hyperexcitability, which triggers the widespread release of tau and amyloid proteins. This flood of tau and amyloid further provokes the brain's microglia, driving them into a hyperactivated state. These newly hyperactivated microglia strip even more extracellular matrix from surviving cells, restarting the entire cycle. This closed loop transforms the disease from a linear progression into a self-sustaining biological cascade. Once this loop is closed, merely clearing upstream proteins like amyloid is no longer sufficient. The cascade now supplies its own forward pressure. Clinical intervention must shift its focus. We cannot target dead neurons. The structure of this transition forces a pause between removing the guard and killing the neuron, giving medicine a definitive timeframe to act. Structural rescue, restoring the integrity of the net, can bring a dormant cell back online. Alzheimer's has a measurable clock, and its middle phase has a distinct structural address. By targeting the protective net, we locate the most critical phase of the disease, the interval where we can still intervene before the threshold is crossed.